Hello and welcome everyone to the Consciousness and Reality Inaugural Colloquium for this academic year. I am Kunal Mule, an astrophysicist and the co-founder of IMIX, which is the Institute of Mind, Intelligence and Consciousness Studies. The CNR Colloquia have brought together researchers from Caltech, MIT and Stanford over the past 2 years. And this year we are welcoming members from UC Berkeley and the University of Arizona to participate. This colloquium series seeks to inspire interdisciplinary investigations on topics such as mind, intelligence, cognition, consciousness, and the nature of reality. So just some quick points before we introduce our colloquium speaker. Selected questions from the Q&A box will be taken after the talk, so I encourage all of you to submit your questions through that Q&A button that you can see at the bottom of the Zoom window. And we will be making this a little interactive and fun. So you can click on the poll button below, and I'm going to launch this poll. Let me see if I can do it now. Okay. So there are two questions which are binary choice. First is, do electric fields create consciousness and mental activity? Yes or no question. And the second question is, which of these hypotheses on the origin of life on the Earth do you find most viable? A is life began in deep sea hydrothermal vents, or B, panspermia, that is life originated elsewhere and came to the Earth. So I will announce the results of the poll at the end of the talk. And I now request our co-host Christoph Koch to please introduce our speaker, Nick Lane.
Thank you very much, Kunal. I would like to introduce our speaker, Nick Lane from London. Nick, I first encountered Nick through some of his books. He's a beautiful, very compelling writer at the intersection. That's also where his professional work has occurred, at the intersection of three fields, which is biology, where chemistry and biology meet, but then mixed in with evolutionary theory. In other words, when you have these biochemical systems and they compete for resources over billions of years, what you get. And he's going to tell us about that. And he's written, I believe now, 5 beautiful books I can warmly recommend. The first book I encountered was Life Ascending, the 3 great inventions of evolution. It's really a wonderful book, really very thoughtful. And his latest book is on chemistry. Transformers, although as far as I understand, the transformers are not like the transformers in ChatGPT. Transformers, the deep chemistry of life and death. He lives and teaches at the University College in London, where he's a professor of evolutionary biochemistry. Nick, welcome to our seminar.
Thank you very much. Thank you for the very kind introduction. Let me, can you see? Can you see that full screen now? I'm assuming you can.
Yep, looks good.
Yes.
Okay, good. Okay, so, well, thank you very much as well for the invitation. It's an honor to give a talk to this audience. I should say at the very beginning that I am not a neurologist, and although I think any biologist worth their salt, any scientist worth their salt should be interested in consciousness. It's not something that many people from the field of biology or evolutionary biology have really considered to be a question in their line. I came to it, as Christoph said, through writing, and I kind of came up against a wall until relatively recently, and I'll tell you how I found a possible path through that wall. But I need to give this disclaimer at the beginning. I'm not a neurologist, and I won't be talking about the brain very much today. So what I will be talking about is what feelings are. And this has become known, as you, I'm sure, all know, as the hard problem in consciousness. And maybe the best framing, the most famous framing, goes back to Thomas Nagel. An organism has conscious mental states if and only if there is something that it is like to be that organism, something that it is like for the organism. And Darwin also made a point which I think is an important one, and I think is basically true, that there's no fundamental difference between man and animals in their ability to feel pleasure and pain, happiness and misery. So these are fairly simple feelings. I'm not trying to claim that animals are capable of sophisticated emotions or really much beyond that, but that basic barrier between information processing and what feelings might be, I personally take it to be quite widespread in animals, as Darwin did. So David Chalmers framed the hard problem. How can we explain why there's something it is like to entertain a mental image or to experience an emotion? It's widely agreed that experience arises from a physical basis, but we have no good explanation of why and how it so arises. Why should physical processing give rise to a rich inner life at all? It seems objectively unreasonable that it should, and yet it does. I probably should apologize to Christoph. I didn't know he was introducing me, but this is perhaps an important slide anyway. There was a bet in 1998. Please correct me, Christoph, if I get anything wrong here, but between Christoph and David Chalmers for a case of wine that within 25 years researchers would discover a clear neural pattern underlying consciousness. And in 2023, after those 25 years, my understanding is they agreed that it's clear that things are not clear, at least not yet, perhaps would be a final clause from Christoph on that.
Also from Dave, he also agrees in this question will have an answer, but we're not there yet.
Not there yet, but the bet has been renewed for another 25 years, I understand.
Yes.
I think, I mean, one of the reasons that I've been drawn back into this, having always had an interest, is I've found myself doing various podcasts and things and being asked about consciousness and thinking about it more. And I think in part it's the rapid development of artificial intelligence, artificial general intelligence. And a lot of people who are working in that field are coming to wonder, what can biologists tell us about the physical basis of feelings? What do we actually know or don't know? So if we have this question, I have this question at least, whether or not feelings as such are an emergent property of neural networks. I think this is the general prevalent view in the AI community. If they're not an emergent property of that, then AI would not have feelings equivalent to animals, even if it's embodied and self-aware. If it is an emergent property of that, then of course embodied AI would develop a consciousness and feelings. And so it's really it's a serious and important question, and biologists ought to be involved in asking it, because biologists more than anybody else study what the properties of life actually are. What do we know about these things? It struck me in thinking about it that a lot of morality really is grounded in the likelihood of suffering, whether we're causing pain to animals, whether even things like religion and suffering on the cross and so on. So if AI does not suffer but merely simulates suffering, then its moral compass will be not remotely human, and it will be much closer to a philosopher's zombie. And that's rather a disturbing thought, and something which is by no means original to me, but certainly it seems to be something to wrestle with and think seriously about. As Christoph said, I wrote a chapter in this book, which is 15 years ago or so now, called Life Ascending, the 10 Great Inventions of Evolution. And you can't write a book with a title like that and not include consciousness. So that was in chapter 9. And I think that the 9 other chapters, I came to some kind of an answer. I won't say a final answer or a correct answer necessarily, but a coherent answer that can explain, in principle at least, the outlines of the problem and the possible solutions. In the chapter on consciousness, I have to say, I really didn't. I think I wrote 10,000 words that said, well, gosh, this is a hard problem and I really don't know. And perhaps moreover, I'm not really convinced that anybody else quite knows either. And I think that's, again, in the terms of that bet, it's more or less says the same thing. Here's a quote that I had in that book. It's easy to imagine robots with the brain power of Deep Blue, that dates it, with language, with senses of the external world, with a near infinite memory. but with no consciousness, no joy, no sorrow, no love or sadness of parting, no exaltation of understanding, no hope, faith, or charity, no thrill of a delicate scent or lightly glancing flesh, no warmth of the sun on the back of your neck, no poignancy of the first Christmas away from home. Perhaps one day a robot will feel all this in its cogs, but for now, we don't know how to program poignancy. No doubt this will come up later on. I'm not sure if it's changed so much since then. What I went on to do in that chapter was to argue that surely Chalmers' hard problem is actually a problem in biochemistry. I would say that as a biochemist. For how does the firing of neurons generate a feeling of anything? How do calcium ions rushing through a membrane generate the sensation of red or fear or anger or love? And I still think that's true. This is a question that if we can't answer it in biochemistry, then I'm not sure that we really understand the question yet. So a little framing for you now in relation to consciousness and reality, as Kunal had asked me to do. This is very much from a biologist point of view. So from my point of view, feelings evolved by natural selection. What that means is that they have selectable utility. They're useful for something, and they have been selected for precisely because they're useful. So utility means there's a survival advantage to having feelings. And conversely, if the optimal response is the same in the presence or absence of feelings, then there can't be a selective advantage. So if the optimal response is to change gene expression or to do something with metabolic regulation or whatever it may be, and that optimal response is the same in the presence or absence of feelings, then there's no benefit to having a feeling, and it could not have been selected. So there has to be something which is which we can see in simple systems that offers a selective advantage, and I'll come to that at the end of my talk. In terms of realism, it seems to me that threats in the environment or opportunities in the environment may be imperfectly measured, but the physical constraints are real, and so feelings are shaped by life and death over generations, by selection over billions and trillions of generations. And if feelings evolved by selection, then they must in some sense be real. There must be some kind of physical state that is selectable. There has to be something there, and in principle, then we should be able to measure it. And this is where we face the difficulty with neural nets, because measurements of conscious versus non-conscious processing in neural nets are really quite ambiguous. So I think it's reasonable to ask the question, is that the right level of analysis? Now, obviously, in some sense it is, but in some other sense, it's getting underneath it all as to what is actually a feeling. I'm not sure it is. I think in terms of information processing, it's exactly the right level of analysis. In terms of interpretation of what feelings might mean in a context, it's exactly the right level of analysis. But in terms of that underlying sensation of hunger or of fear or whatever it may be, I'm not sure it is the right level of analysis. And you could say there's not very much information as such in something like the toothache. I'm actually pulling out an example from one of Christoph's recent abstracts. It can take over your entire being, but there's not much information in the toothache. So feelings then, from my point of view, they're about the subjective living state in relation to the environment. How am I doing now in relation to the world that I'm in? And that's really a property of life. It's not, to my mind, a property of rocks or of the sun or of inanimate objects. And it's not necessarily a property of all of life, but certainly any even single-celled organisms that are dealing with trying to understand their state in the world, there's the potential that there are feelings linked with that. So I had, after writing that chapter in 2009 on consciousness, I kind of stepped away from it all. I read more for that chapter than anything else. I got thoroughly confused by it. I had more angry emails from people telling me I was an idiot and I didn't understand anything. And I thought, oh, this is just not a path that I'm going to go down. Until a few years ago, when I was visited by this guy, Luca Turin, who is a biophysicist. He's somewhat controversial for some of his views on the sense of smell, for example, and I thought that he would want to talk to me about that. But actually what he wanted to talk about was that anesthetics affect mitochondria. And I've spent most of my career actually working on mitochondria and energy generation in cells. Mitochondria are the power packs, if you like, in cells that are generating energy for us. I'll come on to that. And I had no idea that anesthetics had an effect on mitochondria. So that was a revelation to me, and opened a door of a possible path in. Now, Luca Turin is a very pithy writer. A few quotes from some of his papers. He says, almost the only thing we know for sure about consciousness is that it is, so to speak, soluble in ether, chloroform, and other solvents. And he also says, this is a rather nice way of justifying research on things like fruit flies. He says, while it's not known to what extent fruit flies are conscious, they are most definitely unconscious when exposed to chloroform or ether. And some of the work that I'll show you that we've been doing in my lab recently is on fruit flies. Another point that he raises, and it's well known, but I didn't know it, is that anesthetics work on protists, which is to say single-celled organisms. And Luca Turin draws attention to this very wide range of agents, ranging from the element xenon to steroids, can act as general anesthetics on all animals from protozoa to man. Protozoa, of course, are not animals, they're single-celled protists, suggesting that a basic cellular mechanism is involved. Now, that again draws me into the question, because I know nothing at all about central nervous systems, but I do know a little bit about cells and think about cells, and I find that much more of a manageable way of getting at the question. And this last point in yellow, biological activity unrelated to structure, this very wide range of different structures that anesthetics have would suggest that they would interact with different receptors and things like that. And yet the actual loss of consciousness under anesthesia suggests some kind of unitary mechanism, that they're all doing exactly the same thing in rather a similar way. And you wonder how many different receptor interactions could bring that about. It suggests there may be something else going on, which is not really about physical interactions between molecules and receptors that bind to them. And that, of course, is the kind of the explanatory paradigm of molecular biology. It's all about molecules interacting with each other in that way. And so I think anybody who talks about fields, for example, or really, frankly, physics in biology, tend to get a little bit of a difficult ride from molecular biologists who have an unspoken assumption, I suppose, that everything in life can be explained by molecular biology. I don't think that's true. So the idea that Luca Turin has been working up to with Ron Neyman, who's a physicist in Tel Aviv, I think, is chiral-induced spin selectivity. Now, this is a field of physics that's been growing over the last 10 years or so. And there's plenty of examples of how this works. But essentially, my understanding as a non-physicist is that what's going on is that if you're transferring electrons through a chiral medium, and initially that meant chiral molecules adsorbed onto the surface of an electrode, for example, then you polarize their spin in the direction of travel. Now, what was interesting is that there is Luca Turin had picked up a signal in respiration that seemed to suggest that there was some effect on spin related to oxygen, and oxygen itself is a biradical. Let me just put my cursor on here, meaning it actually has two unpaired electrons. Actually, this diagram is incorrect. It should be a single bond here, not a double bond. So there's a single bond and two unpaired electrons in triplet oxygen, and what that means is it must accept electrons in the opposite spin state. And so this rather beautiful and simple idea is that as you transfer electrons to oxygen in the process of respiration, you polarize their spin so that oxygen is more able to accept them. And what anesthetics do is disrupt that spin polarization, so oxygen is now receiving electrons in random spin states. And that slows down respiration. So it's rather a beautiful, rather simple idea, and it would be nice if it was true. But the bottom line is it can't possibly be true because respiration is more complex than that. And to understand why it can't be true, I'm going to have to go into a few of the details of how respiration actually works. So these astonishingly beautiful things, these are the mitochondria that I've already mentioned. These are, I mean, they're usually just called the power packs or powerhouses of cells. This is where cell respiration is taking place in your body now, specifically in these membranes. So this is a mitochondrion. These are the Christae membranes inside the mitochondria. And if you were to...
What's the spatial scale?
So this is around about 1 micrometer in diameter or thereabouts. They can be very different in size. They can be up to 10, 15. You can form effectively tubes, networks of these things, but if they are kind of this kind of mitochondria, which is the classic textbook kind of mitochondria, anything between half a micrometer and a couple of micrometers in diameter. So these, pardon?
But here they're concentrated, right? The density seems to be completely packed.
Yes, they can be quite concentrated in some tissues. I'm not sure exactly what this preparation was here, but they can be very concentrated in tissues, yes. But this may have been something of a mitochondrial isolation. I'm not absolutely sure. It's just a stunningly beautiful picture that I like to show. But this is where respiration is taking place in these membranes. If you were to iron them all out, you'd have about 4 football pitches worth of membrane in the human body. And there's an electrical charge on these membranes, which I'll explain in a moment. But essentially, it's not very much because the distances are very small. It's in the range of 150 to 200 millivolts, but it's across a membrane. This is 2 membranes that you're seeing here. Each membrane is around about 5 or 6 nanometers in thickness, and that means that if you were to effectively shrink yourself down and stand next to those membranes, the field strength that you would experience is about 30 million volts per meter, and that's equivalent to a bolt of lightning. So you have in your body effectively 4 football pitches worth of membranes with a charge across the entire surface area equivalent to a bolt of lightning, and that's what's keeping you alive. And here's roughly how it's working. This is a kind of 101 on basic cell biology, but we're burning food. Let's say glucose, we're breaking it down. Don't worry about the names of any of these things, but to a small 2-carbon molecule called acetyl-CoA. The CoA is bigger than 2 carbons, but the acetyl is 2. And it's spinning around this thing called the Krebs cycle and pulling out CO2, which we breathe out, and hydrogen. The hydrogen's attached to other molecules, but again, that's not the point. It's basically hydrogen atoms, 2 hydrogen atoms. They're split into the electrons and the protons. And we have effectively a current of electrons inside this membrane flowing to oxygen. So the membrane, it's a fatty membrane. There's very large proteins embedded in it. I'll touch on how these electrons are transferred, but in effect, there's a current of electrons in this insulated membrane to oxygen, and that current powers the extrusion of protons across the membrane. That's giving you this, what's called the proton motive force. Effectively, you have a difference in proton concentrations or a difference in pH, and you also have a difference in charge because they all have a charge of +1, and those combined forces give you what's called the proton motive force. Protons come back in through the ATP synthase, driving ATP synthesis, but there's all kinds of other types of work that this proton influx, the proton motive force, can drive. So the simplest way to imagine it is as a hydroelectric power scheme. So the membrane is equivalent to the dam. The proton reservoir, if you like, that's being pumped across the membrane is obviously equivalent to the reservoir. And then the turbine, the electrical turbine that's powering work, well, it's almost literally the case in biology. This is the ATP synthase that I just mentioned. It's a rotating nanomotor that sits in the membrane. It's continually turning at a speed of around about 500 revolutions per second. It's extraordinarily fast. And this will be important later. It tends not to be a single protein like this, but to form dimers that bend the membrane. This is the membrane you can just see here behind that. So these dimers are bending the membrane, and that's actually turns out to be very important, and I'll come back onto that in a little while. So what's pumping the protons out? I mentioned that there are these these proteins, giant proteins sitting in the membrane. Here's one of them. This is the membrane down at the bottom here. This is called complex 1. And it's the protons are being pumped in this membrane domain from the matrix side across to the Christae lumen, but the electrons are being transferred over here. These are iron sulfur clusters. You can see them on the right-hand side here. Most of them are what are called 4Fe, 4S clusters. So there's 4 ions in there and 4 sulfurs. And the distance between these things is usually in the range of 10 to 15 angstroms. The numbers in brackets are from edge to edge. The other numbers are from center to center, and these are in angstroms. And electrons are moving by quantum tunneling. That's generally accepted. Goes back actually to Britton Chance, I think, in the 1960s or thereabouts, from doing experiments at very low temperatures, -70, and the speed of electron conductance at -70 degrees is exactly the same as it is, say, room temperature. So it seems to be quantum tunneling between these things. But there's been big questions in the field that no one has ever really answered. Why are there so many of these things? You could say it's simply to cover that distance, but there's no reason why the hydrogen, the NADH, couldn't bind down here somewhere. You don't necessarily need to have 9 iron sulfur clusters in there. So what are they doing? No one has ever answered that in a very pleasing way. But the other thing to realize is if this is where, if what they're doing there is spin polarizing electrons, if their purpose is to produce a long enough distance that you can spin polarized electrons as they're being transferred, when they get to this molecule here down at the bottom, the quinone, which is binding right in the middle of this molecule here, then any spin polarization is lost. You're back to electron pairs when you're adding them on a quinone to make a quinol. So there's all kinds of peculiarities that we don't understand very well about this membrane complex. So this is a table of other people's work. The references I'm not showing you, but if you're interested to know what they are, I can dig them out and show you. This is from a paper that was just recently published. And much of the work that's on here is from the labs of Phil Morgan and Margaret Sedensky. And what I want you to notice from this is that complex 1 that I've just been talking about, these are various models, some in humans, some in mice, some in C. elegans, some in Drosophila, and so on. And where there's a blue box here, this is with isoflurane or halothane, where there's a blue box, that means that these animal models or patients with mutations in complex 1 are hypersensitive, or at least more sensitive than usual, to anesthetics. So there's quite a lot of different mutations in different animal models where there's a mutation in complex 1 and the result is some level of sensitivity, oversensitivity to anesthetics, meaning you're more likely to potentially die on the table during an operation. Now down here, these are other respiratory complexes in the respiratory chain, mutations in complex 2.
But these are not lethal mutations, right? So people even...
No, these are people... They're not lethal, no. They're people, I mean, people have mitochondrial diseases. They may be lethal, but they're not necessarily lethal. This is a whole field of study. They're extraordinarily complex conditions and with a whole spectrum from lethal within months of birth through to kind of late acquisition in your 50s or 60s. A lot of diseases of aging as well, including things like diabetes and cancer, can be linked to mitochondrial mutations. So they by no means are they necessarily lethal, but if you've got a mitochondrial disease and you're having a general anesthetic, then it will be a good idea that the anaesthetist is aware of that and that they are cautious and careful, in more so perhaps even usual, in giving an anesthetic. It doesn't mean they shouldn't have an anesthetic. It just means that you should be careful.
Thank you.
But down here, all these green ones, this is where there's no effect. So although this is a fairly small sample size and it may simply be noise that we're looking at here, there are really no known examples of mutations in other parts of the respiratory chain, this chain of proteins that convey electrons to oxygen. There's no other mutations, no mutations in these other complexes that seem to cause sensitivity to anesthetics. It's only in complex 1. So that got us interested and quite excited, and I'm going to show you a little bit of our own work, which has been done in fruit flies by Bella Peng and Enrique Rodriguez. And so we were interested to see, do they really affect complex 1? Can we tell anything about this? At this point, we're simply doing some fairly simple respiratory measurements to look what's happening. Now, what we're seeing here is when we give substrates which are effectively passing electrons into complex 1, and we give anesthetics, and we're giving repeated doses of anesthetics, this line going down is showing that we are inhibiting respiration to around about 50% inhibition after 3 doses of anesthetics. This is with isoflurane, and this is with a different anesthetic called sevoflurane, which is slightly less potent here. And then the line which is going up, I've mentioned a couple of times the electrical charge on the membrane, the electrical membrane potential. Interestingly, it seems to go up, not hugely, but it goes up in both cases. And this is just statistical significance. So we see a suppression of respiration. That's to say, a suppression in the current of electrons going to oxygen. And you would have thought that that would lead to effectively less pumping and therefore less membrane potential, but actually the opposite seems to happen. It seems to go up. So something else is happening. We started to dig into what might be happening, and one of the things you can do is inhibit the ATP synthase, this amazing rotating nanomotor. So this is roughly recapitulating what you've already seen, giving multiple doses of anesthetics. Then we inhibit the ATP synthase, and what happens is respiration collapses, because now you can't, there's no more movement of protons going through the ATP synthase. That means you now have all the protons stuck on one side. You've got a strong membrane potential. You can't pump against it, so you can't transfer electrons. The whole thing grinds to a complete halt, and respiration collapses if that's what you do. And what we see then is an increase in membrane potential. That's why you can't pump. But that doesn't actually, this is the membrane potential at the very beginning before we've done anything very much. As soon as you start respiring, it goes down. It gradually, the anesthetics rise it again, and on oligomycin, it goes back up to where you started. So it's suggesting that there may be a problem, a change in the ATP synthase. So we looked at ATP synthesis. This is before you have an anesthetic, and this is afterwards. So this is, again, live in the tissue, the same time that we're measuring the rate of oxygen consumption. And what we're seeing here is a collapse in ATP synthesis. It goes from we're making ATP and positive values up here to we're burning ATP. So what you need to realize is this motor, this ATP synthase, it's completely reversible. It's normally, if you've got this driving force of protons on one side of the membrane, it will force this motor to rotate in a particular direction. But if that potential goes down, then it simply reverses direction. And now what it does, instead of making ATP, it burns ATP to pump protons. So that's what we're seeing here. We're seeing the ATP being hydrolyzed, being burnt to pump protons. That's why the membrane potential went up, and we see exactly the same thing with both anesthetics. And this is, I'm not going to go through the details here, but this is just looking at the production of what are called free radicals, reactive oxygen species. Some anesthetics can cause a little bit of damage, and one of the reasons for that is that they seem to increase the rate of reactive oxygen species flux, and that's what we're showing here. This dip at the beginning is actually an artifact, so we're not really sure. We weren't really sure how to get rid of the artifact, so we simply showed the data with it in. But we do see a clear increase afterwards, and a less clear increase in the case of sevoflurane. So what all of that says then is that we inhibit complex 1 with anesthetics. It had basically no effect on the other respiratory complexes. So we really are seeing also, as the literature says, we're seeing an effect on complex 1 and nothing else. It collapses ATP availability, and that could be insofar as anesthetics affect primarily mitochondria. They do other things as well, no doubt. But insofar as they affect primarily mitochondria, the collapse in ATP availability could explain the downstream effects of anesthetics. It could simply be what Luca Turin calls a synaptic power failure. In other words, you simply don't have enough ATP, enough energy to drive all the work that neurons have to do. And so you're going to see these downstream power failure effects. But there's still a problem here that our work hasn't got at, and nobody else's has, to my knowledge, either. Mutations in the other respiratory complexes, which would also disrupt electron flow to oxygen, which should also collapse ATP availability, should have exactly the same effects. They don't. There's no evidence that they do. Perhaps when we have more data, we'll find out that actually they do. It's just that we've been very unlucky and not seen it yet. But there's another possibility, which is that if the effect is specifically a complex one, and if these ideas from Ron Neyman and Luca Turin are right, that actually anesthetics are scrambling this spin polarization that's taking place in complex one, then the question is, why would you go to the trouble of spin polarizing electrons in complex one if the thing that happens next is you lose it again? It's not carried through anywhere else. So it's really pointing a finger now, specifically at complex one, and saying there's an effect of anesthetics here. It's something to do with spin polarization. So what could that do? Well, the only thing it obviously does is generate magnetic fields. And so I'm kind of forced into a corner, and we have to wrestle with the possibility that there's something to do with magnetic fields and mitochondria. So here we need to, again, take a slight diversion into how mitochondria actually work. And if you are familiar at all with mitochondria, you'll probably have seen this kind of image of what they look like. So what we're seeing here is an outer membrane surrounding it, and then this kind of invaginated inner membrane. And these invaginations are what's called the Christae space. And this is where protons are being pumped. So the respiratory chain is sitting in this inner membrane here. Electrons are being transferred to oxygen. That's powering the extrusion of protons into this intermembrane space between the outer and the inner membrane, and into the Christae lumen here. So there's a relatively positive charge in this space, which I'm showing as red, and a relatively negative charge, which I'm showing as blue. So this is a kind of the classic textbook diagram of mitochondria. And I think one of the reasons it stayed that way for so long is that even people like me can do it in PowerPoint. It's kind of not easy to draw a mitochondrion unless you have some artistic skill. But actually, the reality is closer to this. And it's I apologize for my PowerPoint diagrams. They're still pretty naff, but it's the best I can do. Here's the picture I showed you before of the ATP synthase bending the membrane. We now know that these form dimers, and they sit at the end of the Christae and they bend that membrane like this. And so we have at the end of all of these Christae ATPase dimers. And the pumps that are pumping protons into this space are sitting in the sidewalls here. And the Christae are closed. There's a complex down here. I'm showing just as a black circle. That's called the Mykos complex, sometimes OPA1. These are just different names for proteins and things. But basically what it's doing is closing this space. So it's not generally connected to this intermembrane space at all. Sometimes it is, sometimes they open up. I think they're very dynamic structures. And frankly, this is just beyond the resolution of super resolution microscopy. If we're looking at tissues and the behavior of mitochondria in living tissues, we can see these things in fixed tissue with cryo-EM, tomography. We can see it. We can see it obviously with electron microscopy, but that's dead tissue that's been fixed. With living tissue, with super resolution microscopy, we can see it in isolated mitochondria, but they get damaged. And we can see it in HeLa cells, for example, that are growing in culture. But they don't really need their mitochondria very much, and they behave quite oddly. So if we want to know what's happening in neurons, nobody has ever visualized exactly how dynamic these Christae structures are. It's on the edge of what can be done right now. So this explains a few things. When we're looking at membrane potential, we're very often measuring it using dyes, using what are called lipophilic cations. That's to say, it's lipophilic, it dissolves in lipids, and so it will cross these membranes. Why it would then accumulate in the aqueous phase has always been slightly baffling to me. But it doesn't have to cross. This is a positively charged molecule. It doesn't have to cross this positively charged space here, because actually the positive charge is down inside these closed Christae instead. So it can be drawn by the negative charge inside the matrix, and end up accumulating in the matrix. And that's what you see at the right-hand side here. So this is the nucleus of a cell in blue, staining for DNA in blue. And all of these, I mentioned that they can form long tubules and things that are effectively reticulated networks. this is the mitochondrial network in a cell, and they can really travel quite long distance as they're actively fusing together and fissioning apart. It's a very, very dynamic system. These are not just sitting there in a stationary way. They're changing and moving around all the time. On a matter of seconds to minutes, this is changing all the time. And so this is a dye which has been taken up, which is by the positive charge of the dye is being drawn to the negative charge of the matrix, and it's accumulating there and fluorescing. And so this is how we visualize the membrane potential. And what all of this means then is that we actually have a proton circuit that's structured in space. The protons are being pumped across here. They're coming down inside the Christae. They're coming back through the ATP synthase and back around. So at the very least, we've got a lot more structure in here than perhaps we had appreciated 10 years ago. And the structure means that we have structured movement of charge. And already, as soon as we're seeing moving charge, we can begin to think about fields. So I showed you this slide already, and the question is, well, what's happening here as the electrons are being transferred? And a couple of things to draw your attention to. These iron sulfur clusters are more or less perpendicular to the surface of the membrane. This is an L-shaped molecule, which is unusual in itself, and so the electrons are coming in. And the time for this is around about 100 microseconds is the time that it takes electrons to leave FMN at the top here and arrive down on the quinone. So it's a fairly short period of time, and 2 electrons follow that path, not exactly simultaneously. So there's a lot of things that we don't know about this molecule, but the L shape and this long span and the fact that it's perpendicular to the membrane are all quite interesting. If we've got parallel membranes, and what I'm showing with the arrows here are the movement of electrons towards the surface of the membrane, and we have multiple complex 1 molecules sitting on this flat surface, and we have multiple parallel flat surfaces, and this I'm showing the same dimension in each case. These are all moving into that surface. They should generate a magnetic field for a short period of time. And the protons are coming down inside here towards the ATP synthase dimers, and in this one in the opposite orientation in the other way. That should generate magnetic fields at right angles perpendicular to that. And so there should be some synchronization. Now, as soon as the electrons have arrived here, there are also complex 1s on the other side as well. And they, as soon as the field has dissipated on one side, they should be now able to go the other way. And so we can go backwards and forwards, and we're going to be generating, if we have synchronization of electron transfer, we're going to have an oscillating magnetic field generated by mitochondria in the direction, pointing in the direction of the Christae membranes themselves, and the whole thing synchronized together by fields generated by the proton flux within these Christae, synchronizing the different Christae within a mitochondria. Now, there's some, let's say, weak but beautiful evidence that there may be something in this. So these are mitochondria, actually, I think in heart tissue in this case, but these are the Christae membranes that you can see. This is a mitochondrion here. This is a separate mitochondrion. This is the outer membrane between the 2 mitochondria. And you can see that the Christae membranes are lined up from one mitochondria to the next one. And it's repeatedly so. You can see it in this case as well. You can see it here. You can see it there. It's remarkable that these Christae membranes are lined up. Now, this is work from Martin Pickard and Doug Wallace from some years ago now. But they were also able to move, to sever the links between, any physical links between these mitochondria and move them apart slightly. And you could see as they come back close together again, then the Christae will line up again. If they're further away, they're not. So there's a space factor going on here. As soon as I showed this picture to a physicist, they said, "Oh, those patterns remind me of the magnetic strip domain structure of metal films, which you can see by magnetic force microscopy, and this is what they look like." And yes, it's probably a bad idea to suggest that it looks a little bit similar. That's probably not a very helpful thing to say, but certainly at the very least, there are larger effects here that go across between mitochondria, which are not explained by simple molecular biology and by binding of these mitochondria, there's a larger scale fields operating here. And I don't think it's an electrostatic field, because if it was an electrostatic field, these things should be offset against each other. Here we have a positively charged lumen of a Christae abutting another positively charged lumen of a Christae in each case. So they're not offset against each other. That suggests a magnetic field rather than an electrostatic field, which is generating that effect. So it's an obvious question then, how strong could these fields be? And I'm going to hop very quickly over this, because I've just checked the time and I'm maybe taking a little too long over this talk. So I'm going to speed up a little bit. Perhaps we can come back to this, but effectively field strength is the current over 2 pi r, and we've calculated the number of electrons conducted across complex 1 is about 10 nanometers is that distance. We've converted it into Tesla. The number of complex 1 molecules per cell, this is a relatively low estimate. In fact, it's probably closer to 1 million. And so if there's 1,000 mitochondria in a cell, 1,000 complex 1 molecules per mitochondrion. If there's 10 mitochondria in a synapse and 25 Christae per mitochondrion, then there will be 40 complex 1 molecules per Christae, 20 on either side of the Christae. Current flows for 100 microseconds from NADH to the quinone. In each 100 microseconds, 2 electrons are transferred across the 20 complex 1s per Christae, with 25 Christae and 10 mitochondria. And the current then, as a quick calculation of the current, over what radius? So over 1 micrometer, the field strength would be something in the order of 300 microtesla. If we're looking at 5 micrometers, it would fall to about 60 microtesla, which is roughly the Earth's magnetic field. So we're above the Earth's magnetic field, which I'm treating as a kind of as a background cutoff, that if we're not stronger than the Earth's magnetic field, then there's a question as to whether or not it would be sensed. But we know that plenty of organisms can sense the Earth's magnetic field. So if these fields generated by mitochondria over cell-like distances 1 to 5 micrometers is stronger than the Earth's magnetic field, so there's no reason why it shouldn't give rise to these kind of strengths. The oscillation frequency, assuming the complete cycle of turnover of complex 1 is in the region of 1 to 10 milliseconds, so the actual time of electron transfer is only a part of that cycle, and that gives an oscillation frequency of 50 to 500 microhertz, which is going to depend on things like the membrane potential itself. But at the low end of that, 50 microhertz, we're of course in the gamma frequency range, which is interesting at the very least. So neural mitochondria in principle could generate an oscillating magnetic field with a frequency of 50 to 500 hertz and a field strength of about 300 microtesla over 1 micrometer. These are back of the envelope calculations, but they show in principle that it's possible that mitochondria can generate magnetic fields. What could they possibly do? Well, that's an interesting question. I mean, in principle, they could exert effects on voltage-gated channels. There is some work that I'm citing in this slide here, going back to the 1990s, showing that influx of calcium, for example, into lymphocytes, it can be altered quite substantially by oscillating magnetic fields in that kind of range. So I think it was 120 microtesla and 50 hertz, 150 microtesla maybe. So also people like Joe Kirschvink have called attention to magnetite crystals, and there are supposedly 100 million of these of single domain magnetite crystals per gram of the human brain, more in the brain stem by the sound of it than elsewhere. I've seen papers as well saying that people with Alzheimer's disease have more of these things, that they're kind of smooth on the surface, which suggests pollution. So it's possible that they've got nothing to do with physiology at all. Though I think in some fish that they have been shown to affect magnetic sensing of the Earth's magnetic field. So it's also possible that they do have physiological effects, and they are associated with membranes. So there are things that fields could do, magnetic fields could do in neurons. Also, if they were going to exert effects, then I mentioned already that they should be along the plane of the Christae, and here we're seeing mitochondria always with their Christae pointing at synapses and at the plasma membrane over here. So if they are producing fields, it's interesting again. There's some movement in the Christae orientation here, but the ones that are closest to the membrane are usually pointing at the membrane, and I've heard this said as well for auditory nerves. And we know generally that mitochondrial potentials could be linked with the EEG. All kinds of mitochondrial diseases cause EEG abnormalities, and many mitochondrial diseases are associated with epileptic seizures and so on. And my understanding is we don't really know what's producing the EEG. We know that it's networks of neurons depolarizing. You need hundreds, if not thousands of them to produce the kind of voltages that we're reading. But I just want to leave you with this thought that mitochondrial membrane potential is about 200 millivolts, whereas it's only about 60 millivolts for neuronal membranes. And there's a lot more mitochondrial membranes in parallel than there is on the synaptic membrane. So it's quite possible that a lot of the voltage changes that we're measuring with the EEG are actually mitochondrial voltage changes and not just the plasma membrane. So I just want to finish in the last five or 10 minutes or so to ask why. I hope I've got 5 or 10 minutes. I'm slightly over. I apologize for that. But just to finish, if electromagnetic fields are indeed generated by mitochondria, are indeed central to consciousness, then why mitochondria? Why a little bit inside a cell? Why would that matter? Why should we care? Why would you want to believe anything I'm telling you about mitochondria? The critical thing is that mitochondria were bacteria once. They were free-living bacteria that were acquired by a host cell, and there's strong evidence, and I've done quite a lot of work on this over a couple of decades. They were central to the evolution of eukaryotes via an endosymbiosis. So the cell that acquired them was quite a simple archaeal cell without very much going on inside in terms of morphological complexity. Mitochondria today are well known to be a stress hub in cells, and we need to realize that they are actually the interface. Although they're buried inside cells, they're the interface between the cell and the environment in terms of substrates, in terms of electron acceptors, so oxygen, where we're burning food, if we have food, metabolic poisons, if we have cyanide or anything like that, then this is the place where it is first felt. It's the mitochondria which are the interface with all of these important aspects of the outside world. The way I've been thinking about it, I won't go into details here, but the free energy principle from Carl Friston, which I'm sure many of you will be familiar with, cells are in effect minimizing free energy, or what I think Mark Solms has called felt uncertainty, by active inference, by some kind of Bayesian best guess as to what state a cell or a nervous system or whatever it is wrapped in the Markov blanket, whatever its state may be. Now, I find it easier to think in terms of cells rather than in terms of complex central nervous systems. And what I'm going to put to you in the last 5 minutes is that a feeling is an electromagnetic state, the kind of thing I've been talking about, corresponding to a metabolic perturbation that tells an organism how it is doing in the world. And I'm just going to go through this for the last few minutes. So here's a kind of my picture of a bacterial cell. Outside, stuff is happening. Inside, we have metabolism. Yes, of course, there's genes and things as well, but in terms of immediate reaction to the outside world, the only thing that's happening inside a bacterial cell is metabolism. We can see the plasma membrane as a Markov blanket, and it's translating, just as the brain does, whatever stimuli are happening in the outside world into a different language. And that other language inside a bacterial cell is metabolism. There's nothing else going on in there. And metabolism, well, it's about, in a bacterial cell, is 10 billion reactions, 1 to 10 billion reactions every second. It's a colossal number of reactions. And you can wonder what would keep all these reactions in the same kind of phase space. Why would they be doing the same thing? And part of the answer to that, I've shown you already the Krebs cycle earlier on, is generating a membrane potential, and that membrane potential is pushing back. It's continually pushing back. So we're burning substrates to generate a membrane potential, and that membrane potential is driving metabolism. So in other words, metabolism generates a membrane potential which drives metabolism. There's a beautiful circularity to this, and it means there's a push and a pull in this system which is in series linking it up. Now, it's not just one Krebs cycle. There are potentially 10,000 different Krebs cycles all spinning at the same time in the same cell, all contributing to this membrane potential. So when we're talking about 1 billion reactions every second, the membrane potential really is giving you an indication of what is the overall state of the cell as an entity. It's the plasma membrane, remember, in bacteria. So as a single entity, as a self, and all of these constituent billion reactions a second, the plasma membrane is giving you a kind of a synchronized readout of the state of metabolism at any one moment. So why would that matter? As I said at the very beginning, a feeling needs to be useful for something. And so what would this be useful for? I think the answer relates to motility, almost certainly. I mean, if you think about if your metabolism is pushed into a bad place by a difficult environment, and the only way you have to fix it is to change the activity of genes or something like that. There's no obvious reason why, in the presence or absence of a feeling, you would have any difference in the response. If the only thing you can do is change your gene expression, then there's no benefit to attaching a feeling to that or not attaching a feeling to that. But if you have the ability to move, if you've got a flagellum here and you can move, then you need to make a snap decision as to, am I going to stay here or am I going to go? Am I going to activate my flagellum and leave? Or should I stay here and weather it out? You need to make that decision before you've activated all this allosteric response, because the allosteric response is effectively going to mask the situation that you're in. You make it slightly better, but now you don't know how bad it was anymore, and you don't know what your reserves are and what your resources are and whether you can weather the storm or not. So what you need is a kind of a snapshot readout of your state and some kind of way of deciding whether or not you can cope. You specifically, with your life history, do you have the resources and the wherewithal to deal with this threat that you've got, or should you activate your flagellum and paddle away? So it's decision making, it's agency, it's self, all these terms that we might talk about in relation to consciousness, they're all implicit in how a bacterium is going to behave. I'm not going to go through this in the context of things, but the ratio of the electrostatic field to the electromagnetic field generated by the movement of charge, we can think about the balance between the two, giving you slightly different, effectively, structures of water bound to enzymes and overall physical state of the cell. In terms of, we can distinguish between substrate deficiency, low oxygen or heat, or normal conditions like growing. I'm not going to go through that, but I'm happy to come back to it if anybody wants to ask any questions. Of course, we are eukaryotic cells. We have multiple mitochondria. They became incorporated in. These mitochondria need to talk to each other, and they need to talk to the plasma membrane. And at the moment, we don't really have a very clear idea of, do they use fields to do this kind of thing? They're all affecting metabolism in the middle. It could be all molecular biology that's doing that, but I just want to leave you with this short video, which I think that molecular biology at the moment can't really explain what this cell is doing. This is a predatory ciliate lacrimaria olor. It's stunning, it's beautiful. Molecular biology certainly can explain the neck extensions with the cytoskeleton and so on, but this is going to very particular places. That's not just kind of grasping around trying to work out, is there some food here or whatever it's doing. It's very particular in where it's going. It knows somehow exactly what it's doing, and I don't think that we have an explanation for that behavior yet. It's picking up on charge or on fields or on something or other, in my opinion. And I have a feeling that the future of biology at this point is understanding the physics of biology rather than the chemistry and the molecular biology, and understanding how organisms really behave as an entity, as a self, even at the level of single cells. Right, I have to thank my lab. This is the wider lab, many of whom work on the origin of life and origin of eukaryotes and questions like that. You'll all know that I've only come to questions of anesthetics relatively recently, but it's been great fun. And thank you. I'm very happy to take questions. I'm going to stop sharing now.
Thank you very much, Nick, for that fascinating talk. Christoph, since you need to leave, I'm going to give our co-host a chance to speak quickly if there is a question or comment. Christoph.
Yeah,
please.
I mean, the two obvious questions come to mind. So, there are mitochondria throughout the body, just not in our brain, right? But the liver or the kidney, as far as we can tell, including the heart, are not the organs of consciousness. In other words,
I agree, yes.
You can get a transplant, a heart transplant or other transplant, and you're still the same conscious mind. And B, even within the brain, so we do know the cerebellum, right, where 80% of your neurons are, the little brain. If you lose it, if you never had it, like agenesis of cerebellum, or you lose it because you had tumor there, etc., patients never complain about loss of consciousness. They don't complain that their world looks different, etc. But again, these Purkinje cells have huge number of mitochondria. So
yes,
what's
so
that all about?
the simple answer is I spent quite a lot of time talking about parallel Christae and the number of mitochondria, and they have to line up. And so a lot of the time that simply doesn't happen. So you would not be generating a coherent field if the orientation of the Christae is different, or if there are not enough of them there. So you would need a certain number with a certain orientation. What I imagine in my mind is that the Christae are waving around, and occasionally, if you have, for example, a depolarization of the plasma membrane, of the axonal membrane, or the dendrite membrane, then they're going to snap into some kind of parallel formation and generate a field. And that field is telling you about the state of cells. Now, it's not going to be all cells. It's going to be, in the case of the brain, specific cells which are linked to certain homeostatic mechanisms of one sort or another. I'm thinking mostly in terms of interoception rather than exteroception. But again, in terms of which bits of the brain, we don't know at the moment. I mean, I'm partial to the views of people like Mark Solms, who talk about the brainstem and the reticular activating system, in terms of where attention is and the basic feelings that are underpinning it in relation to interoception in the body. So those are the places that I would look to see, do we see mitochondrial morphology where they're lining up and the Christae are lining up and we have enough of them? So I'm not.
But they are lined up in the cerebellum, right? They are like, because you have these parallel lamellae, right, in the cerebellum, these two-dimensional slabs. So they're really, I mean, that's a remarkable thing about the Purkinje cell. They're these beautiful, like books on a shelf. They're really aligned. Anyhow, it's just a thought. And so you're saying that in the other organs, the mitochondria simply aren't aligned in the right way.
Exactly. Yes. I mean, I think when you think about what is required here, the last thing that a multicellular organism needs is for its kidneys to be having feelings. You need them to be at the level of the organism itself. So with a eukaryotic cell, you need to be, the self is the cell as a whole. You don't want the mitochondria to be having feelings. If it's a bacterial cell, the bacteria needs to have some kind of an explanation of its environment. So a bacterial cell may have feelings, but mitochondria, which derive...
Well, okay, but I assume the same mitochondria will generate the same magnetic field in kidneys.
No, because the orientation is different. If you look at kidney mitochondrial Christae membranes, they're all over the place. They're jumbled up. There's you know, insofar as it generates a field at all, they all cancel themselves out. You need a very specific orientation of Christae. So that's the easiest way to test this initially, rather than trying to measure fields. As an orientation of Christae that are required for this to work at all. So the simplest way to disprove it is to disprove that the Christae ever line up in that way. But the pictures I showed you, and I'm aware that I'm kind of cherry picking pictures that help me make my point, but there aren't so many out there. I mean, if I could find a lot more pictures of mitochondria with Christae at wrong angles or right angles, I would find them. It's work we need to do, I think.
All right. Thank you very much. Beautiful talk. I learned a lot about mitochondria I didn't know. Thank you.
Thank you.
Thank you, Christoph, for joining.
Thank you
and
for coming at all.
We have several questions in the Q&A box, but before we go to those, I'm going to reveal the results of the poll. So I'm going to press share here, and I hope you're able to see this on your screens. 103 participants took part in this poll. And first question, do electric fields create conscious and mental activity? 67% answered yes, and 33% no. Second question.
I wonder if that would have changed after the talk, or if it's gone down.
Perhaps, yeah. Second question was, which of these hypotheses on the origin of life on the Earth do you find most viable? 82% answered life began in deep sea hydrothermal vents. So they've all read your works, Nick, and they're all convinced. Yeah.
You didn't give them the choice of terrestrial geothermal systems, which would be most chemists' preferred answer to that question. So,
right. Next time, maybe we'll give
maybe
them
a few
those.
people funneled into that answer.
Right. But 18% answered the other part, which was panspermia. Okay, so I'm going to stop sharing and we'll move on to the Q&A very quickly. The first question is from Professor J. McClelland at Stanford, from psychology, of course. This is fascinating stuff, taking me deep inside tissues. But we are still left with the problem of experience, are we not? Part of the question for me is, where is the experiencer if these tiny fields are the things experienced? And there are a few more questions that echo that sort of feeling. And Akanditi Das here says, fascinating demonstration of magnetic fields generated by mitochondria, and perhaps this has a relevance to the effect of anesthetics and perhaps even states of consciousness. But how do they give rise to actual felt experiences and qualia? What's your
response? So the way in which I have tried to think about that is not in relation to the central nervous system at all, but in relation to bacteria, which will seem probably very bizarre to most of the people who think about consciousness at all. And of course, I have no idea if bacteria are actually conscious. So the first thing is qualia is what I mean by feelings, but I'm not really talking about qualia in terms of why we experience what we do in the color red or whatever it may be. I'm thinking about the most basic feelings that relate to living. So things like pain or hunger or whatever it may be. Now, the question is, I suppose, I mentioned early on, there has to be a physical state. If it's under selection, there has to be some kind of physical state. And that physical state is not obviously to me at the level of neural networks. Obviously, they embellish that state in some way, but I think it's there at the level of bacteria. So the question then is, how would you distinguish? And I'm saying it's the physical state of the cell, which is to say the fields will organize. And I hesitate to say this, but I can't actually think of a better explanation. The balance between electrostatic and electromagnetic fields influence the structure of water within a cell. Now, that doesn't mean to say I'm a homeopath or something. What it means is that the water is structured on the surface of membranes, water is structured around ions, water is structured around the surface of proteins, and cells inside them are gel, and most, if not all, of the water is structured. But it's not completely structured. You can also see cytoplasmic streaming. There's movement in there as well. Now, if you impose electrostatic fields across cells, then you start to break open the clusters of water that are surrounding ions, for example. You're going to reorientate them in the direction of the charge of the field. And if you're oscillating that field, you're going to keep on constantly breaking hydrogen bonds, breaking up clusters, and that's going to release ions into the system. So effectively, you're going to make the cytoplasm more reactive. You're going to free up the space to get to enzymes. You're going to change that system in a blanket way across cells if your balance is towards electrostatic fields. If your balance is towards magnetic fields, then you tend to have the opposite effect, which is to say you tend to enhance the clustering of water molecules around things. So what I'm imagining then is a feeling is really very much the physical fabric of the cell, the state of the cell. That's what a feeling physically is. It's the if you like, the elasticity of the physical elasticity of the cell. A simple way of looking at it to see if there's any truth in it would be to measure very simple things like the speed of cytoplasmic streaming. Does that change in response to magnetic versus electrostatic fields and so on? Do we change the structure and the elasticity of water within cells? So that's my attempt at an answer. That's not an answer at the level of the human nervous system. That's an attempt to say a feeling is a physical state, and that physical state tells you about your state in relation to the world. It's not a random state. It says, if I am in this state, I am in trouble for this reason. And if I'm in that state, I'm good for that reason. And so there's a simple association between physical states and potential outcomes.
Thank you. Here's a question from William Newson. I'm just going to shorten it up because we're short on time. And how exactly would mitochondrial EM fields account for the experience of pain which is one example that you gave, which mitochondria matter, the molecules in the nociceptors, in central pain pathways, in cells all over the body? How do the presumed mitochondrial effects interact with standard pain sensing and transmission pathways?
I'm not going to give a good answer to that because I don't work on any of those things and I don't really know the anatomy of the central nervous system or exactly how pain works. What I would say is that there would be, my understanding with phantom pain and things like that is there would be certain populations of neurons that are telling you that you are in pain. And you may or may not actually really be in pain. So it would be the mitochondria in those neurons that are misbehaving for whatever reason and telling you that. So you would need to be looking at the centers and the activation of those centers. So I mentioned Mark Solms's work and the reticular activating system and attention and so on. I imagine there would be a problem there in terms of attention. I imagine that those deeper seated feelings and emotions that's where I would primarily look for changes in mitochondrial behavior in relation to things like pain.
Thank you. Maybe
I'm not trying to suggest that this is separate from neurotransmitters. It will be linked with neuromodulators, neurotransmitters, and all the rest of the wealth of the central nervous system. Is simply that underpinning. I suppose the thing is that there's no reason why serotonin should be associated with positive feelings and acetylcholine, for example, with anxiety or whatever it may be. You could reverse those round in principle. There's nothing about the chemical structure of a neurotransmitter that would say this should have this association with that. What we need, I think, in terms of understanding the relationship between the actual real world fact that you're doing badly in relation to this environment, that it's painful, whatever it may be, it is a physical state that's telling you that, which you can't divorce from your circumstances. So it's I'm kind of several levels down on the pitch of that question, but I think that still there would be this relationship between a population of mitochondria that are telling me my physical state is bad, and that's linked to then the neuromodulation and neurotransmitters and the CNS and the rest of it.
Very nice. We'll end with this one question, which is quite interesting, because here's a person who says that if feelings are in an electromagnetic state, I can see the future where we could be able to physically replicate these states, perhaps in silicon. I'm an electrical engineer, but I'm still very interested in what makes human intelligence different from AI. I'm also interested in augmentation for human intelligence. How can someone like me who have such a background help with furthering this kind of research? So firstly, and of course, the related question is that, do you think there would be non-biological life forms, right? Or let's say, for example, silicon.
Yes, I mean, I certainly think there can be non-biological life forms. I have a real problem in defining what life is or could be, and so but I think the more interesting question is, would things like feelings emerge from a sufficiently complex set of neural nets? And the answer that I've been putting to you, I suppose, is no. So why not? Well, because number one, it's not specifically about information processing so much as coming up with a readout of your metabolic state. And there isn't an equivalent in neural nets of metabolism where you've got a wet system. It's a kind of metabolic wetware with 1 billion reactions every second, and a requirement to have some sense of what are all these reactions doing? What state am I in? You know, so a second after second, another 1 billion reactions going on. It's incomprehensible what that means and how you keep a handle on it. So I think that's something which is very much a property of wet matter, and the way that life is grounded in chemistry and the speed of these reactions and the speed of the enzymes and the structure of water around enzymes and so on. That's not to say that that's the only way it could be done, or that would be the only benefit for it. But I do think that we have some kind of an obligation as biologists to try to figure out what do we think is happening at the level of living things. And then we can think, well, would that apply to some kind of embodied robot? I think the answer is no. Someone like Mark Solms would say the answer is yes. I think at the very least, I can't pretend to know the answer, but I think at the very least we need to recognize that life has got a certain set of problems. It's also generational. Selection is over billions of generations. It begins to associate certain states with certain outcomes. Some of them do better than others. And those states are really wet-ware metabolic states of disruption from homeostasis. And it's not obvious to me that any of the things I just said really have much of an equivalent in terms of how AI works.
All right, very nice, Nick. I think we've run out of time. But just a quick announcement, the next colloquium in the Consciousness and Reality series will be in January, so stay tuned for more details on that. And finally, I would like to thank Nick Lane again. And Nick, kudos to you for being one of the very selected few biochemists in the world doing some research on consciousness. Would like to also thank our co-host Christoph Koch, all organizers and participants who made this event happen. Have a great day, everyone. Take care.
Thank you very much.
Thanks.